Deep beneath the skin's surface lies an intricate extracellular matrix dominated by collagen, the structural protein responsible for firmness and elasticity. Beginning in our mid-20s, endogenous collagen synthesis gradually declines by approximately 1% annually. Concurrently, environmental stressors such as ultraviolet radiation and cellular oxidative stress accelerate the degradation of collagen fibers through enzymatic pathways like matrix metalloproteinases. As this structural scaffold weakens, fine lines and dermal thinning naturally emerge over time. While cosmetic interventions focus primarily on surface appearance, dermatological research increasingly investigates how systemic nutrition influences cellular integrity from within. Can specific dietary compounds support dermal structure, or is skin aging purely dictated by genetics and chronological time? To understand the boundary between marketing claims and biological reality, we must examine the biochemical machinery that governs skin degradation and repair. At the cellular level, collagen synthesis depends heavily on dermal fibroblasts, specialized cells responsible for assembling amino acids into stable triple helix protein structures. This complex biochemical process requires specific cofactor nutrients, particularly vitamin C, which enables the enzymatic hydroxylation of proline and lysine residues. Without adequate cofactor availability, stable structural collagen cannot effectively form. Furthermore, dietary polyphenols and carotenoids found in nutrient-dense plants act as biological antioxidants. Research indicates these bioactive compounds may help neutralize reactive oxygen species generated by solar radiation and natural metabolic processes. By attenuating inflammatory signaling cascades, such as the NF-κB pathway, certain phytonutrients appear to reduce excessive enzymatic matrix degradation, potentially helping preserve existing extracellular collagen networks over time. Thus, nutrition operates as a steady supply of structural precursors and cellular protective agents. To evaluate potential benefits, scientists evaluate research through a clear hierarchy of evidence. In vitro laboratory studies demonstrate that isolated polyphenols can shield cultured human dermal fibroblasts from oxidative damage. However, isolated petri dish environments do not replicate human digestion, hepatic metabolism, or microvascular delivery. Moving higher in the evidence hierarchy, observational population studies consistently associate Mediterranean-style dietary patterns, rich in vegetables, fish, and healthy fats, with greater measured skin elasticity and lower markers of photoaging. Randomized controlled human trials provide further clarity. Several clinical trials investigating hydrolyzed collagen peptides suggest that daily oral consumption may support skin hydration and dermal density over 8 to 12 weeks. Nevertheless, researchers caution that study parameters vary, and further large-scale human trials are required to establish standardized guidelines. An essential consideration in nutritional science is bioavailability. How efficiently ingested compounds are digested, absorbed, and delivered to dermal tissue. Intact collagen molecules are too large to be absorbed directly. Digestive enzymes break them down into individual amino acids and small peptides. Whether circulated peptide fragments preferentially target dermal tissue or general bodily protein repair remains a subject of ongoing investigation. Furthermore, dietary benefits operate within biological limitations. Excessive intake of refined sugars can trigger advanced glycation end products, cross-linking collagen fibers and rendering them rigid and fragile. Unprotected exposure to ultraviolet light and chronic systemic stress can easily outpace the subtle protective effects of dietary bioactives. Therefore, dietary support must be viewed not as a standalone solution, but as one integrated component alongside environmental protection. Ultimately, skin vitality reflects broader systemic physiological health. Current scientific consensus indicates that no single nutrient or isolated supplement can independently prevent structural aging. Evidence suggests that a holistic lifestyle strategy, combining a balanced, plant-rich dietary pattern with daily broad-spectrum sun protection, restorative sleep, physical movement, and effective stress management, provides the most meaningful support for long-term dermal health and systemic longevity. By focusing on overall lifestyle consistency rather than isolated quick fixes, we support the body's natural regenerative capacities over time. Disclaimer: this documentary is for educational and informational purposes only and does not substitute for professional medical advice, diagnosis, or treatment. Always consult a qualified healthcare provider regarding personal health conditions.